That is used for the classifications. Now, if we take about the molecular approach, how we can characterize these proteins. There are certain molecular approach to characterize the BT proteins. So based on different molecular approach like protein profiling, plasmid profiling, total DNA profiling. Okay. So these are the technique. Protein profiling can be done by using first you have to isolate the protein. First we can isolate the protein from the from the different bacteria. Then we can use different techniques like protein profiling. Protein profiling is done through the HDS page. Then by using different uh, technique like Malditov and other proteins, 2D and 3-dimensional gel electrophoresis, we can characterize the protein, how much size is range of that particular proteins. Then plasmid profiling also done by the agrogel analysis by comparing with the other standards proteins. And total DNA profiling also be done same as a plasmid profiling. So if we talk about the location of these proteins, so it because this is a bacteria, Bacillus thuringiensis is a bacteria, and the bacteria has a plasmid as well as their chromosomal DNA, plasmid as well as chromosomal DNA. So plasmid may be large size or small size. So if larger two type of plasmid are present on this MBT, Bacillus thuringiensis, large plasmids, small plasmid. In case of large plasmids, majority of the cryogenes are present on the large plasmid. Majority of the plasmids are present on the large plasmid. While in case of a small plasmid, the smaller plasmids they do not contain much on the smaller plasmids. If you talk about the chromosomal DNA, this chromosomal DNA contains the vegetative insecticidal proteins BIP, that is known as BIP. Chromosomal DNA also contain different insecticidal protein, that is vegetative insecticidal protein BIP. Now, target specificity of certain protein. If you talk about the different target of different class of the proteins, which or insect order is the targeted for? Cry1 Lepidopteran, Cry1 Lepidopteran, Cry2 Dipteran and Lepidopteran, Dipteran and Lepidopteran, Cry3 Coleopteran, Cry1 Lepidopteran, Cry2 Dipteran and Lepidopteran, Cry3 Coleopteran, Try to A. Dipteran and Lepidopteran. Try to miss in group of Clitus, Dipteran and Lepidopteran. Try 3 Coleoptera. Try 4 Diptera. Try 4 Diptera. Try 5 Nematodes. Try 5 Nematodes. Try 5 and 6 Nematodes. Nematicidal activity. They have been well characterized for these target orders. Cry 7 Coleoptera, Cry 8 Lepidoptera, Cry 9 Lepidopterans, Dipterans, Cry 12, 13 nematicidal activity, Cry 14 again Nematodiptera, Cry 16 Dipterans, 17 Dipterans, 18 Coleoptera. So these different class of the cry proteins are have a specificity against different insect order. It's a long list you can see here. So cry 42 you can cell killing toxin genes, cytocidal human cells, cry 46 cytocidal human cells. So these are the various cryogene which have been other further they have different subclasses. You can see here. So I am not going into the detail all these things. If we talk about the classifications, as we now discuss, what we will look five different most important classes which we are using nowadays in the different crop species. Okay, so that we will see cry 1, 2, 3, 4, and 5, and their size of protoxin, size of the toxin, or size of the proteins, the structure, what structure is there, size of active toxins, sector specificity, or which order, percent homology in between that. So if you take about the Cry1 protein, Cry1 had different subclasses like A to K. Cry1A, Cry2V, Cry2C, likewise. Cry1A, Cry1V, Cry1C, Cry1D, Cry1E, till Cry1K. So they have the different subclasses based on their some different physical and chemical properties. They have been subclasses. If you take the size of protoxin means the inactive form because the newly synthesis protein, that is protoxin, but that is inactive. Okay, that the size varies from 130 to 160 kilodalton. 130 to 160 kilodalton. One protein size is 130 to 160 kilodalton protein is there. Okay. Next is Structure of this scribin A protein is 65. Structure is bipyramidal. Structure of scribin A is bipyramidal. Size of active toxin is 60 to 70. Active toxin is 60 to 70. Insect specificity against the Coleoptera, Lepidoptera, and Coleoptera. So this scribin group of the proteins is specific to the Coleoptera and Lepidoptera. Coleoptera and Lepidoptera. And percent homology, if you talk about the percent homology, is 80 to 90%. 80 to 90% homology is matching with this. Percent. 80 to 90%. 80 to 90% is matching. Next is Cry2 protein. Cry2, they have subclasses A, B, and C. A and B and C. The size of this protoxin is 70 to 71 kilodalton. 70 to 71 kilodalton. Okay. Structure is cuboidal shape. Structure is cuboidal shape. Size of toxin is 65. 65. Lepidoptera and Diptera. Insect specificity, Lepidoptera and Diptera. Lepidoptera, Lepidoptera and Diptera. Percent homology 80 to 90%. Percent homology 80 to 90%. Try third. If you take the example of try third protein is subclasses A, B, C, and D. Size of protoxin 73. Size of protoxin is 73. Structure is rhomboidal bipyramidal. Rhomboidal bipyramidal. Size of active toxin is 55. Insect specificity Coleoptera. Insect specificity Coleoptera. Percent homology is more than 75. Percent homology is more than 75. Try4 again has subclasses A, B, C, and D. Protoxin 20 to 140 kilodalton. 20 to 140 kilodalton irregular. Structure is irregular, say 55 is the active protoxins. Insect specificity Diptera. 29 to 55% of human. Cry fifth and sixth. Cry fifth and sixth. 81 to 2. 81 to 2 kilodalton. 81.2 kilodaltons. Size 40 to 60 kilodalton. Larvicide of Lepidoptera, Diptera, and nematocytes. So this picture represents the evolution of the new crygenes, how the new crygenes get evolved by the rearrangement between the different class of the domains. So we know very well the cry proteins have three different domains, domain 1, 2, and 3. New proteins, they rearrange between those different class of the cry proteins, then the new crygenes get evolved. Now, if you see the BT formulations, there is third generation of BT formula nowadays we are using. That is first generation, second, third generation. It's a first generation product, a blend of spore and crystal, which form a native strands. Form a native strands. Blends of the spore and crystals we are using. Second generation product, genes which coding for delta endotoxins. In case of second generation, we were used about the genes coding delta endotoxin from the several strands. Third generation's product, dead recombinant Pseudomonas fluorescent cells. Pseudomonas fluorescent cells, which VT formula depend on the pest to be controlled and the pest and the plant. Depend on pest to be controlled and the plants. We were used the particular BT. We have to prepare a particular BT formulation used on the control and plants. Active ingredient in commercial formulation is spore and crystal complex. Spores and crystal complex. So you can see here the formulation is emulsion. Formulation type is emulsion type. We have applied to the different agricultural crop and forestry purpose. Emulsion form of formulations we are using for the agriculture forestry purpose. Encapsulation type agriculture and forestry. Encapsulation type agriculture and forestry. Vegetable powder for the garden and agriculture. Garden and agriculture. Granule agriculture and forestry. Granules agriculture and forestry. If we take the example of powders, powders like forestry for the forestry. Now, advantage and disadvantages of the BTA toxins. Advantage is safety. It's a safe against a human health. Specificity. That is if we take the example safety purpose, so let me tell you about effect on human health. Effect on, we will see the safety aspect of the BT technology. Now, if we talk about the effect of BT on the human health, BT protein, because human beings are the non-target organism. Currently, we are talking about the effect of BT on the non-target organism. Specificity of BT for its target insect is one of the most characteristic feature, which is the ideal method for the biological pest control. In fact, different strands of BT have specific toxicity to certain target insect. The specificity rests on the fact that toxicity of the BT protein is receptor mediated. Because it is directly not acting on any target organism, it is based on the receptor mediated. If receptor is present, then only this protein, BT protein, will interact with the target insect. Otherwise, it won't interact or won't affect adversely affect that growth. This means for an insect to be affected by the BT protein, it must have a specific receptor in the gut where the proteins can interact or bind to them. Fortunately, humans and other majority of the beneficial insects do not have these receptors. Okay, so there is no question about the effect, adversely effect of the BT on the human. Similarly, if we talk about the BT crops which are placed on the market, they must pass through a stringent regulatory test. Okay, before releasing into the market any BT crops, so they must pass very stringent regulatory tests, which include those for the toxicity and allergenicity. Toxicity and allergenicity. The U.S. Environmental Protection Agency has already administered toxicology assessment, and BT protein have already been tested even at relatively higher doses. According to the Extension Toxical Network, pesticide information project on several universities in the US, there is no complaints were made after 18 humans had 1 gram of commercial BT preparation daily for 5 days. Now, if you see the effect on environment or soil ecosystem and groundwater, the BT protein is moderately persistent in soil and classified as immobile, as it does not move or leach with groundwater. It does not move or leach with groundwater. It does not particularly persist in acidic soils conditions where exposed to sunlight. It rapidly destroys due to UV radiation. Rapidly destroyed due to UV radiation. There is independent experts which have conducted study to investigate the impact of BT crop on soil organisms. Other insect species that are considered beneficial in agriculture. There is no adverse effect has been found on non-target soil organisms. Even when these organisms were exposed to quantity of BT far, then what would actually occur under natural crop growing conditions? Likewise, research done by US EPA Environmental Protection Agency revealed there is no change in the soil microbiota. Next is animals and insect. If you take the safety aspect of animals and insect, on different tests were conducted on the dog, guinea pig, rat, fish, frog, slender, even birds. The BT protein was found not to have any harmful effect. It is not what is that no toxic effect were found on beneficial predatory effect because there is large number of the test has been done against the different class of the animals like guinea dogs, pigs, rats. So not even a single organism saw the harmful effect or adverse effect on their growth and development by the this BT. So in 1999, there was a report that pollen from the BT corn had a negative impact on the monarch butterfly. That was the report in the 1999. Okay. This report raised a concern or question about the risk of BT crops on the non-target organism. A recent study on the same experiment showed that BT corn poses negligible threat to monarch butterfly in the field. There is a collaborative research effort by the scientists in the US and Canada. They have produced informations to develop a formal risk assessment of the impact of BT corn on monarch butterfly population. They concluded that in most commercial hybrids, BT expression in pollen is very low, and laboratory and field studies show there is no acute toxic effect at any pollen density that would be encountered in the fields. We can take the several advantage of the BT crop BT crop in the recent studies. Biodegradable. So this another one is biodegradable. This BT protein is again it's not as I told you, it's a very it's biodegradable. Biodegradable is not sustained for longer period in the environment or soils. Okay, so it's a can be degraded after some time. Potential there is large number of potential for the different target insect and pest. Disadvantage, it's rapidly inactivated by UV light, heat, and extreme pH. If you provide the high light condition, UV light condition, heat, and extreme pH, it gets rapidly degraded by the environment. Susceptible to protease in the leaf exudates. Easily removed from the plant surface